Good morning, everyone. Thanks for being here. I am excited to introduce today's speaker, Dr. Mackenzie Kelgard. She joined my lab in November, so she's still pretty new. We pretty much threw her into the deep end of a pretty big microbiome selection experiment, and she has done wonderfully, so we are definitely really happy to have her. So hopefully at some point she'll be able to give a seminar again on some of the cool stuff she's going to be doing here at Purdue, but today she's going to be focusing on some of the work that she did for her PhD. So she got her BA at the University of Denver, and then very recently finished her PhD at Texas A&M. So Mackenzie, I will hand things over to you, and we're looking forward to your seminar.
Awesome, thanks, Laramie. Let me go ahead and share my screen. All right, figured out technology. All right, cool. Well, thanks everybody for being here today. Yeah, like Laramie said, I'm going to be mainly talking about some of my dissertation research that I did at Texas A&M. And then at the end, I'm going to touch on, what I'm doing here as a postdoc at Purdue. So one of the major questions of my research has been what drives species abundance. And there are, of course, a number of factors that can influence the abundance of a species through space and time, one of those being resource use. So Charles Elton, who's known as the father of ecology, was one of the first to kind of formally propose this relationship between resource use and species abundance in what is now known as the Eltonian pyramid. And this is the idea that organisms at the base of the food web will be more abundant than those at the top. Now, another factor that can influence species abundance is competition. And that includes competition both between species or interspecific competition and competition within a species or intra-specific competition. And so I chose to test the relationship between these variables using invasive ants as a model system. And invasive ants are great for looking at the relationship between these variables because they're often hyperabundant in invaded communities. And one of the possible reasons for how invasive ants are able to reach such a high abundance is resource use. So for many invasive ant species, we know that more herbivorous colonies tend to grow faster, workers live longer, and they also forage more aggressively compared to colonies that are more predaceous. So in that way, they really do seem to follow this traditional Eltonian pyramid where colonies that are feeding more as herbivores, and typically that means that they're feeding on higher amounts of carbohydrates or sugar resources in the form of insect-produced honeydew or extrafloral nectar. So colonies that feed on high amounts of those sugar resources tend to reach a higher abundance compared to their more predaceous counterparts. So another factor that's thought to influence the abundance of invasive ant species is their proclivity towards intra-specific cooperation. So for many invasive ant species, they show reduced territoriality and aggression towards members of the same species. So essentially by avoiding these very costly competitive interactions and cooperating with each other instead, it's thought that invasive ant species are able to reach a higher abundance. So I chose to test some of these hypotheses using two invasive ant species. The first being the Tawny Crazy Ant, or Nylandaria fulva. So this ant has become a major pest throughout most of the Gulf Coast states, primarily because it's able to reach these very extreme densities. So this is an example at one of my field sites here. So I've got a pitfall trap there in the center, but I really want to draw your attention to the ground surrounding the pitfall trap. So hopefully this video will work. But you can basically see that the ground is just crawling with Tawny Crazy Ants. And this is really common to see at infested field sites. They're crawling all over the ground, all over the vegetation, up your boots. They're really crawling everywhere. And this ant species doesn't sting, so it makes it a little bit less horrifying, but it's still a very big problem and very difficult to control their numbers. So the second invasive ant species that I looked at was the red imported fire ant, or Solenopsis invicta. If you've lived in the South at all, you're likely very familiar with this ant species because they're everywhere in the South. And I love to include this picture here because fire ants never look happier than when they're stinging someone. And so this can result in a lot of painful stings, kind of a raised pustule. And for individuals that develop an allergy to those stings, such as myself, so I actually develop a pretty gnarly blister when I get stung by fire ants. So that can be pretty bad. So I actually had to carry an EpiPen with me pretty much everywhere that I went in Texas because fire ants are everywhere throughout Texas. So this is a really common sight to see throughout Texas, but all of those raised bumps are all fire ant mounds with upwards of 200,000 to 300,000 workers within a single mound. So there really are quite a few fire ants in this field site. So it's really a terrible place for a picnic. But because they're so abundant, they also have a pretty big impact on other species. So you can see them kind of ripping apart this snake right here. So super abundant. And one way for us to estimate resource consumption is using naturally occurring stable isotopes. So if you'll remember from your chemistry class, the main difference between a light and a heavy isotope is simply in the number of neutrons. And what's great about nitrogen in particular is that the ratio of heavy to light nitrogen, which is expressed in this delta notation, increases predictably moving up the food chain. So essentially by measuring the isotope values of consumers, we do this using a mass spectrometer, we're able to estimate the trophic position of an organism. And so that's usually estimated relative to this kind of traditional scale where a trophic position of 1 corresponds to primary producers, 2 herbivores, 3 primary predators, and so on and so forth as we move up the food chain. So one example of this can be seen in Hellman et al. 2013, and they looked at omnivorous caddisfly larvae and what these larvae were essentially eating in freshwater streams. And they found that this omnivorous species shifts its diet from a more predaceous diet in the wintertime to a much more herbivorous diet in the summertime. So essentially by estimating trophic position on this more continuous scale, ecologists are able to get a better idea of the strength of these trophic interactions. And so although this is a really helpful technique, one problem is that isotopes vary a lot through space and time. And so each location differs in the amount of isotopes inherent in the environment. And this environmental variation is naturally reflected in the tissue, in the organisms that live in those environments. So unfortunately, we're not able to kind of directly compare these isotope values across different locations. So in order to estimate a trophic position, we first need to account for that environmental variation so that we can then compare across these different locations. So kind of the first major decision for ecologists wanting to use this technique is to choose a baseline material. And this is meant to really account for that environmental variation. And when we look in the literature, we actually see a lot of different baseline materials that are currently used. And this can range from autotrophs or primary producers to herbivores to soil to particulate organic matter and so on and so forth. So there are a lot of different baselines that are currently in use. And Cabana and Rasmussen in 1996 demonstrated that estimates can vary by as much as 8 trophic levels, depending on the baseline material that's used. So finding the appropriate baseline material is very important in accurately estimating trophic position. So the next decision for ecologists using this technique is to estimate trophic position relative to that baseline material. And so this can be done, once again, when we look in the literature, we see that authors are employing really a variety of methodologies to estimate trophic position. And that can range from a variety of different mathematical formulas. It can also be done with statistical analyses, compound-specific isotope analysis, which is essentially another technique that measures the isotope values of specific amino acids in the tissues of consumers. Or authors choose to sometimes directly compare isotope values or just kind of ignore the problem. But unfortunately, there's currently no standardization in the literature. And a lot of people are using this. So I found that over 5,000 studies use stable isotopes to estimate trophic position. And each baseline and methodology could generate very different conclusions. And this is important because this technique is currently being used to inform important management decisions, such as fisheries management, the control of important animal and human disease vectors, as well as wildlife management decisions. So it's really important for us to kind of standardize our approach moving forward to make sure that we're generating the most accurate conclusions. So this brings me to kind of the outline of my talk for you today. So I'm really going to first talk about a literature review before I apply that to invasive ant species. And I'm pretty excited that this review was actually recently accepted in Ecological Monographs. So that's pretty awesome. So for this literature review, I had two primary objectives. And the first was to kind of quantify or characterize this variation that's currently in the literature. And then the second objective was really to determine, if this matters. So I wanted to assess the potential consequences. I did this in two different ways. The first way was to look at potential consequences on estimates of individual species. So oftentimes we're using this technique to look at the diet of an individual species in different locations. And then I also wanted to see if there was any consequence on food web structure. So first, I characterize variation in the literature by using the following relevant search terms on Web of Science. And as I mentioned, I ended up with over 5,000 papers, the vast majority of which focused on aquatic ecosystems. So this is actually something that other people have noted in the past, that aquatic ecosystems kind of tend to dominate that stable isotope literature. So to kind of make this a little bit more manageable, I sorted all of those studies by relevance and then reviewed the first 300 studies. So I ended up with 200 studies total, 121 of which focused on aquatic ecosystems, 79 of which focused on terrestrial ecosystems. And then I used log linear models to analyze that data. Okay, so looking at our results, here I have our two ecosystem types, so aquatic and terrestrial ecosystems on the x-axis, the proportion of papers that I reviewed on the y-axis. When we fill in the data, we see significant differences depending on the ecosystem studied. So authors focused on aquatic ecosystems tended to use a single source mathematical formula, whereas those focused on terrestrial ecosystems tended to use statistical analyses or baseline subtraction. And I think it's important to note here that theoretically, all of these methodologies can be applied regardless of the study system. So we're not entirely sure why there's such a difference between these two ecosystems. So likewise, we see something similar with the baseline material. So authors focused on aquatic ecosystems tended to use herbivores as a baseline, whereas those focused on terrestrial ecosystems had none or not specified, often because they were using statistical analyses or primary producers. So next, I looked at the potential consequences on individual species estimates. So I did this in a couple different ways. The first way, I extracted published trophic positions of commonly studied species from the literature. And these published trophic positions were essentially estimated using different methodologies and baselines. And I did this with the help of my former lab mate, Jeremy Hewlett. So we ended up with 834 trophic position estimates of 63 different species from 101 publications. So the second way that I did this is I actually applied the most common methodologies to 10 existing stable isotope data sets. So I wanted to see, do these methodologies generate different estimates when I use the exact same data set? So to do this, I had a single source formula with two different baseline materials whenever possible. And then I had a variety of other common methodologies that are used to estimate trophic position. Okay, so for the results here, I have my published trophic positions by species. So all of the species are here on the x-axis, estimated trophic position on the y-axis. Here I'm filling in the data. There's quite a bit going on in this graph, but I essentially just want to show that there are significant differences depending on methodology. So for example, if we look at the harbor seal or Phoca vitulina, we can see that published trophic positions differ by as much as 2 trophic levels, depending on the methodology used. And so this has a pretty big impact on our overall conclusions because it ranges from thinking that the seal is a tertiary predator going all the way down to a strict herbivore. We also see an effect of baseline material on published trophic positions. So for example, with albacore tuna, we can see a very high trophic position when herbivores or particulate organic matter are used as baseline material. And this drops by almost an entire trophic level when predators are used as a baseline material instead. So when we look at estimates using existing data sets, we see a similar trend. So here, once again, I have species on the x-axis, this time using numbers to designate those different species. I have aquatic data sets on top and terrestrial data sets on bottom. Again, a lot going on here in this plot, but it's essentially just to show that we really see the same thing where there are significant differences in trophic position estimates depending on the methodology. And again, this is really using the exact same data set, but we get big differences depending on what we choose to use. So for example, with the Japanese spiny lobster, we can see some pretty big differences depending on whether, for example, compound-specific isotope analysis is used versus a single source formula. And then we also see differences depending on the baseline material. So when autotrophs are used as a baseline, we get a trophic position of 2 or strict herbivores. But when herbivores are used as a baseline material, that goes up almost an entire trophic level. So yes, looking at this first part here, we do see a significant effect on trophic position estimates for individual species. So next, I wanted to look at whether there was an effect on conclusions about overall food web structure. So to do this, I took those same 10 stable isotope data sets and this time statistically analyzed trophic groups instead of species. So oftentimes, instead of analyzing differences between individual species, ecologists might group them according to their trophic level. So for example, this is data taken from Stefan et al. 2013. Here I have methodology once again on the x-axis, estimated trophic position here on the y. What's particularly great about this data set is that it was a controlled feeding study. So we know that there should be 4 distinct trophic groups. So that includes a plant, an herbivore, a primary predator, and a secondary predator. So when we fill in the data here, again, we see some pretty big differences in the actual estimate. But when we look at statistically analyzing those trophic groups, first looking at raw values, we see 3 statistically distinct trophic levels. Whereas when we look at a single source formula, we get only 2 statistically distinct trophic levels. And finally, for compound-specific isotope analysis, we get our expected 4 statistically distinct trophic levels. So yes, we do see this in most data sets. So that's 7 out of 10. And part of the reason why I think we don't see it in all 10 data sets is due to sample size. So data sets that had the same conclusions collected 3 or more samples per trophic group. So this actually got me interested in this issue of sample size. So I did a power analysis to determine the suggested sample size for each method. So here, methodology on the x-axis, suggested sample size on the y. When we fill in the data, we see that compound-specific isotope analysis requires the smallest number of samples to detect significant differences between trophic groups. And then that can range up to about 5 samples on average for the single source formula, going all the way up to 36 samples on average for baseline subtraction or raw values. But when we look at the actual number of samples that are being collected in the literature, here I have the minimum number of samples collected per trophic group on the X, proportion of papers that I reviewed at the beginning there on the Y-axis. We see that 41% of all studies collected a minimum of only 1 sample per trophic group, and 26% did not specify how many were collected at all. And so we see with this that there's kind of a big need for us to be collecting more samples in all of the trophic groups that we're looking at. So in conclusion, I found large variation in the baseline material and methodologies that are currently used in the literature. And this does have a big impact on individual species estimate, as well as conclusions about food web structure. And like I mentioned, these are currently being used to inform pretty important management decisions. So it's important that we really standardize our approach moving forward. And oftentimes studies, reviews are compiling all of these different estimates, but they're not taking into account the effect of baseline and methodology. So it's going to be important moving forward to account for that variation as well. Some of my recommendations are to complement stable isotope analysis with more traditional approaches, such as gut content analysis or direct observation. But ultimately, we need to be collecting more samples. So ideally 5, but 10 for best practices for every trophic group that we're looking at. And in cases where sample size is unavoidably constrained, such as many wildlife studies, compound-specific isotope analysis may be best because it requires the fewest number of samples. But ultimately, additional experimental studies are going to be necessary moving forward to determine really the best methodology and baseline for each habitat. Okay, so now kind of applying what I just talked about to the Tawny Crazy Ant. So as I mentioned at the beginning, Tawny Crazy Ants are able to reach these very extreme densities. So these are examples of 2 pitfall traps that I leave out at my sites. Left those out for just 24 hours, and I get upwards of 5,000 ants and sometimes even 10,000 ants within a single trap. And that really just didn't even make a dent in the population size at all. And they can be particularly problematic because they're sort of attracted to the electrical wiring. So they can get into electrical wiring, cause it to short circuit. So they can be very costly to kind of repair those damages. And they're also very difficult to control. So I wanted to examine whether there was a relationship between Tawny Crazy Ant abundance and their trophic position. And then I also wanted to see how this might influence their competitive interactions with other ant species. So as I mentioned at the beginning of my presentation, one factor that may promote the abundance of certain invasive ant species is a more herbivorous diet. So colonies that tend to feed on high amounts of carbohydrate resources or feed more as herbivores tend to reach a higher abundance than those that feed as predators. And this also corresponds with their competitive interactions with other ant species. So ants that reach a higher abundance and are more herbivorous tend to outcompete other ant species for resources compared to their more predaceous counterparts. So I wanted to examine Tawny Crazy Ant resource use and competitive interactions in multiple sites in Texas with the expectation that as ants feed more as herbivores, they would be more likely to outcompete other ant species for resources, and this would also correspond with a higher abundance. So to do this, I sampled from 3 main locations in Texas, the first being kind of around College Station, which is right where Texas A&M University is, the second being just outside of Austin, and the third being way down south here, right on the border of Texas and Mexico. And just to give you an idea of just how enormous Texas is, driving from College Station to southern Texas was about 375 miles. So that would be like driving from here to Nashville. And I did that drive every month for a little while in 2018. So I really got a lot of good driving time in, a lot of podcasts and audiobooks. So within each location, I tried to divide those into multiple subsites. So I ended up with 5 total sites. As I said, I sampled there every month from April to August in 2018. So habitats ranged from this very grassy habitat going up to kind of more of a forested habitat. So I laid out pitfall traps as one measure of ant abundance. So you can see an example of that here. It's laid flush with the ground. I laid those out for 24 hours. And then I also used baits to measure ant forager abundance. So I laid those baits on the ground as well as in trees. This is an example of what that looked like. So I had a weigh boat and I laid a hot dog slice as well as some honey, which is kind of the universal ant attractant. And I laid those out for 1 hour. At the end of 1 hour, I counted the number of ants that were there, identified the ant species, and kind of identified which ant species had won the bait. So then using that pitfall trap data and the bait data, I assigned each ant species a behavioral dominance score. And I'm not going to get into too many of the details on how I did this, but I'm happy to talk more about this after the presentation if you're interested. And I did this using a collie matrix. So each ant species was assigned a behavioral dominance score ranging from 0 to 1, with 0 meaning that the ant species had access to the bait, but they lost that bait to another ant species. And 1 meaning that they had access to the bait and they outcompeted other ant species for that bait. I then collected ants, plants, known insect herbivores, and known arthropod predators for stable isotope analysis. I chose to use a single source formula with plants as the baseline material to estimate trophic position, but I also statistically compared that with other known trophic groups, and I paired that with direct observation based on my literature review suggestions. Okay, so looking at ant trophic position by ant forager abundance on baits. So here, once again, my expectation was to see a negative correlation between these variables. But counter to my expectations, I actually saw a positive correlation. So as ants were more predaceous, foragers were also more abundant at those baits. Likewise, when we look at ant trophic position by Tawny Crazy Ant behavioral dominance, we see a positive correlation between these variables. So as Tawny Crazy Ants were more predaceous, they were also more likely to outcompete other ant species for baits. And this relationship is likely due to the relationship between ant behavioral dominance and the number of foragers at those baits. So as there were more ants on the baits, they were more likely to outcompete other ant species for that resource. When I statistically compared Tawny Crazy Ant trophic position with other known trophic groups, we see that Tawny Crazy Ants are omnivorous. So they have a significantly lower trophic position compared to arthropod predators and significantly higher compared to arthropod herbivores. And this is supported by my direct observations. So I commonly saw Tawny Crazy Ants tending a variety of different aphid species. I also saw them on different extrafloral nectaries. So you can see an ant here on partridge pea. And I also saw Tawny Crazy Ants feeding on arthropod prey, both alive and dead. So this is an example of Tawny Crazy Ants that are taking down a cockroach. They don't act very quickly when they're preying on something, but within about 5 to 10 minutes, they had killed this cockroach and were taking it apart. So I do have evidence of them feeding on arthropod prey as well. And this dietary flexibility may help explain the relationship that I found between ant trophic position and their pitfall trap abundance. So this is log transformed number of ants in pitfall traps, and then their trophic position here on the y-axis. So when I fill in the data here, we see no significant correlation between these two variables. So, and this ranges from about 10 ants within a pitfall trap going all the way up to about 8,000 ants within a pitfall trap. So I think that, really by feeding flexibly, these ants are able to kind of have these variety of different densities. So in conclusion, trophic position, counter to my expectations, were positively correlated with ant abundance at baits and their resource dominance. And Tawny Crazy Ants were much more predaceous than I expected. And this can be done in a couple different ways. So Tawny Crazy Ants may increase their prey consumption, which would raise their trophic position. So we know that in other invasive ant species, high worker activity that's kind of fueled by high carbohydrate consumption can increase the likelihood of those ants encountering prey. Or these ants may be decreasing their sugar consumption. So at least in the laboratory, we know for Tawny Crazy Ants that carbohydrate deprivation can increase their aggression. So this may be another reason for why their trophic position was higher than expected. So ultimately, future studies are going to need to kind of look at this to really tease apart why we're seeing a more predaceous diet, essentially. But I think this shows that, kind of the importance of omnivory. So omnivory rather than strict herbivory may drive invasive ant abundance, particularly in the case of the Tawny Crazy Ant. And so we know that top-heavy food webs are not uncommon in nature. And this is kind of, a shift from that Eltonian pyramid to something that looks a little bit more like this, where, upper trophic levels are more abundant than the ones below them. And this happens especially when predators are omnivorous. So I think we might be seeing something like this, especially with the case of Tawny Crazy Ants. Okay, so now kind of shifting focus from Tawny Crazy Ants to the red imported fire ant. As I mentioned at the beginning, one factor that may promote the abundance of invasive ant species is their proclivity towards intra-specific cooperation. So by avoiding these costly competitive interactions and cooperating with each other, it's thought that many invasive ant species are able to reach a greater abundance. And looking at intraspecific cooperation is actually great for fire ants. So fire ants occur in two different social forms, the first being the single queen or monogyne form. So this is kind of the stereotypical ant colony that we usually think of, with just one egg-laying queen within the colony. These single queen colonies are not cooperative with each other, so there's no sharing between different colonies, and workers are highly aggressive towards conspecifics. And if we contrast that with the other social form, or the multi-queen polygyne form of fire ants, this can have anywhere from 2 to hundreds of egg-laying queens within a single colony. So this multi-queen form is thought to be highly cooperative. So there's thought to be a lot of sharing of resources and workers between neighboring colonies. And they're thought to be workers are thought to be not very aggressive towards conspecifics. And this difference in cooperative behavior is thought to correspond with a difference in abundance. So Porter et al. In 1991 found that single queen colonies were much less abundant. So they had about 295 mounds per hectare on average, compared to 680 mounds per hectare on average of multi-queen colonies. So there's quite a big difference in the abundance between these two forms. And it's thought that it's driven by that cooperative behavior. But you might notice that I put a question mark here, and that's because this cooperative behavior between multi-queen colonies is actually poorly documented in the field, except for a single study by Batkar and Vinson in 1987. So I wanted to really examine whether these multi-queen colonies are actually sharing with each other. And I wanted to look at this in two different ways. So first, between colony sharing. So I had the hypothesis going into this that single queen colonies will not share with each other, whereas polygyne or multi-queen colonies will share with each other. And that's based off of those previous assumptions. But then I also wanted to look at within colony sharing. And here, I wanted to see if these multi-queen workers are sharing with all larvae regardless of relatedness, because when you have all of these queens within a single colony, you're oftentimes encountering larvae that may be less related to you. So I really wanted to see what that looks like on within colony sharing. So to look at between colony sharing, I used stable isotope analysis, but this time I kind of tweaked it to use it as an isotope tracer. So isotope tracers kind of take advantage of the fact that these heavier isotopes occur very rarely in nature. So if we spike the environment with an unnaturally high level of that heavy isotope, we're able to trace the movement of the isotope through the environment. So the idea is that if we feed a fire ant colony high amounts of nitrogen-15, we can then quantify resource sharing between that treated mound and any neighboring mound. So the hypothesis was that multi-queen colonies will be sharing with each other. So we detect a high amount of nitrogen-15 in the treated mound, as well as the neighboring mounds. Whereas single queen colonies would really keep that to themselves. So we would only detect high amounts of nitrogen-15 in the treated mound. So to do this, I looked at 6 total sites in Texas. Sites consisted, this is kind of an example of what a site looked like. So I had 3 to 4 clusters of mounds. Here's an example of a cluster where I had a treated mound and then several neighboring mounds at varying distances. So all mounds within a cluster were between 0.5 to 30 meters from the treated mound. And I did this varying distance to really see whether there might be some effect of distance on resource sharing. I also performed a genetic assay to determine the social form of each of those mounds and combined that with population genetics to kind of get an idea of the genetic relatedness between these different colonies. And to do all of that genetics analysis, I collaborated with Dr. Ed Vargo, who's also at Texas A&M University, his postdoc, Dr. Pierre-Andre Ayer, and one of his PhD students, Joni King. So to spike these colonies with nitrogen 15, we fed each treatment mound a total of 160 milligrams of nitrogen 15 labeled glycine, and that was mixed in a sugar solution. This amount was based off of some preliminary tests that we did in the laboratory that I don't go into here, but again, happy to talk about isotopes if you ever want to. And this is an example of what that looked like. So we have the treated mound here with 3 1 milliliter vials that are within 10 centimeters of that treated mound. We put those really close to really ensure that only workers from the treated mound are feeding on a solution. We replace those every other day for a 2-week period. And here you can see an example of fire ants feeding on the solution. So they've actually built up some soil around the lip of the solution, or the lip of the vial, and you can see workers kind of feeding within it. So looking at our results, all treated mounds showed highly elevated nitrogen 15 values by the end of the experiment. So that shows that our methods were highly successful in spiking the values of the treated mounds. So we had 11 single queen and 8 multi-queen treated mounds. And then looking at our results of the untreated mound, here we see that very few mounds shared regardless of social form. So about an equal number of single queen and multi-queen mounds shared with the treated mound, but the vast majority did not share at all. And mounds that shared were likely part of the same colony. And so this is based off of distance. So all of the mounds that shared were within 5 meters of that treated colony. Those that shared also had a very low pairwise FST value when compared with the treated mound. So this suggests that mounds that did share were likely part of the same colony. We know that fire ants are polydomous, so this means that essentially a single colony can have multiple mounds. So this is likely what happened in our results. So next, looking at within colony sharing, I did this research with 2 former students in the Eubanks lab, so Colin McMichael and Dr. Allison Bakoven. So to look at within colony sharing, we established 6 single lineage multi-queen colonies or polygyne colonies. So this consisted, here's kind of an example of this here, where you have a queen and all of her brood. And we wanted to test worker discernment between close siblings or larvae from the same mother and not siblings or larvae from a different mother. So to do this, we labeled brood with food dye. So we had 3 colonies that were labeled with green food dye, 3 colonies that were labeled with yellow food dye. And that dye was mixed in water and artificial nectar, and no proteinaceous food was given to those colonies to kind of encourage feeding on these solutions. And this lasted for 6 days. So you can see an example of what that looked like here with the colony and the different colored food dyes. So by the end of the 6-day period, you could differentiate brood based on these different colonies just with the naked eye. So you can see brood that were fed the green food dye and brood that were fed the yellow food dye. So next, we actually mixed those together to make 18 experimental colonies. And these were paired according to color so that we could really differentiate and determine and really know where that larvae had come from. And each experimental colony consisted of about 120 workers, mixed with 50 larvae from the same natal colony as the workers or close siblings, and 50 larvae from a different colony of the workers or not siblings. So this is kind of an example of what that looked like here, where you have the workers and the differently colored brood that were mixed together. So next, experimental colonies were given nectar mixed with fluorescent dye. So you can see an example of what that looked like here. And what's great with the fluorescent dye is that we can actually use a black light to see the movement of that dye. So you can see which larvae have essentially been fed the fluorescent dye and which have not been fed that dye. And after 18 hours, brood and workers were then separated and counted. So looking at results for within colony sharing, we see that significantly more related larvae remained after 18 hours with the workers. And we searched the inside and outside of the nest, but found no larval remains. So this difference in the number of larvae that remained after 18 hours could potentially be due to preferential cannibalism of the less related larvae. Likewise, we also see that workers fed a higher percentage of related larvae compared with unrelated larvae. So this leads me to the conclusion that polygyne fire ants are not cooperating. In fact, polygyne colonies were just as isolated as monogyne colonies. So that may be suggesting high intraspecific competition, which is what a past study by Weeks et al. In 2004 had found. Our laboratory results indicate well-developed nestmate recognition, so workers really do seem to be able to differentiate between related and unrelated larvae. And this is supported by a past study by Oben et al. 1993. This has important implications for fire ant management because the idea based on those previous assumptions was that if you had a field site of all multi-queen colonies, if they're sharing with each other, then theoretically you would need less insecticide to try and kill these fire ant colonies. But based on these results, these multi-queen colonies are just as isolated as these single queen colonies. So we really need to treat them exactly the same way. This tests fundamental assumptions in kin selection theory. So workers may be increasing their own inclusive fitness by preferentially caring for more related individuals within the colony. So future work should compare with fire ant colonies in other parts of their invaded range to really determine how universal these results are. And if these multi-queen colonies are not cooperating, this kind of begs the question of why we see such a big difference in the abundance between these two social forms. So future work should really kind of try and tease apart some of the reasons for this. So in conclusion, I identified some best practices for stable isotope analysis by demonstrating the consequences of appropriate baseline material, methodology, and sample size in accurately estimating trophic position from stable isotope data. I also showed that Tawny Crazy Ant trophic position was positively correlated with their abundance at baits and their resource dominance, which suggests that omnivory rather than strict herbivory may drive the abundance of some invasive ant species. So we kind of see a higher abundance of these omnivores than some of the lower trophic levels. I also show that multi-queen colonies of fire ants are not cooperating and maybe actually competing with neighboring colonies. So we really need to look at their relationship with that abundance. And as I mentioned, this does have important implications for kin selection theory. So throughout my presentation, I've talked a lot about the kind of interplay between resource use, competition, and species abundance. So for my postdoc research, I'm kind of including an additional factor, that microbes, kind of how that influences species abundance. So this is really, my research is really looking at how microbes might interact with plants. So either in the plant roots or the leaves, to either promote or inhibit the abundance of insect herbivores. So I'm, as Laramie mentioned, I only started in November, so it's relatively new here, but I'm excited to somewhere down the line talk to you about some of that research as well. So with that, I'd like to thank all these wonderful people for their help in making this research possible, as well as my funding sources. And I would be happy to take any questions.
Great. Thanks, Mackenzie. Really enjoyed your seminar. I think I'll just say I really appreciate that you had such a nice framework predicting what you thought you were going to see. And then in the end, a lot of times your data did not support that. But I think what's really cool is that you end up finding out, sort of viewing things a lot differently. And I think the complexity that you sort of unravel is much more interesting potentially than, if you had this clean, beautiful, all the predictions supported type of story. So yeah, so I think,
thanks, Larry.
yeah. I mean, I guess, I try to be positive because that's kind of what happens a lot of times with the microbe work that you're involved in too. So, so we have a couple of questions in the Q&A. So the first one is, could the results of your last experiment be different if the brood were from the same colony but of different lineages?
Yeah, no, that's a great question. That's kind of one of the limitations of, that particular laboratory experiment, because we did keep those colonies separated. So it could have, something to do with environmental odor. But that's really something that I think we really need to look more into. So something that I didn't touch too much on in my presentation, but these fire ants kind of are differentiated by this, difference in allele, these differences in alleles. So we actually use that to kind of determine the social form of these different fire ant colonies. So determining whether it's a single queen colony or a multi-queen colony. And so, part of the hypothesis behind that is if you have the right allele, then you're going to be treated, you're going to have this kind of be treated regardless of relatedness. So that's kind of why we were expecting that in the laboratory colonies, and why we kind of had them separated, because we expected that, regardless of their relatedness, they'd kind of treat everyone equally because they had kind of the right allele. But yeah, so that's definitely something that moving forward would be interesting to look at, of, whether you actually had all of them together, if you see a difference in their behavior. But yeah, good question.
So I guess, yeah, I'll ask a question, kind of tying things into what you're going to be working on now. So where do you, I guess, see stable isotope analysis maybe fitting into adding microbes into the picture? Or do you have just any general thoughts on that, or, what people are doing?
Yeah, no, that's a great question. It's definitely something that I'm thinking about too. But yeah, I think what's so great about stable isotope analysis is that you can kind of see the flow of nutrients in a system. And so something that I'm interested in kind of looking at with the microbes and the plants, of seeing whether there's an exchange of nutrients. So, we see that in some examples where microbes are kind of providing these nutrients to the plant and they're benefiting the plant in that way. And then there's kind of other ways where microbes are kind of priming these plant defenses. So it's not even necessarily an exchange of nutrients, but they're kind of helping the plant by turning on those plant defenses in advance of the insect herbivores getting there. So that's where I think it could be interesting to kind of incorporate stable isotope analysis into that, to really see whether it's benefiting the plant by different, bring nutrients in, or if it's something else, and kind of pair that with some other techniques to really look at that. So yeah, so I think it could be kind of fun.
Yeah, definitely. So I guess, yeah, if anyone else has any other questions, feel free to type them in the Q&A.
Laramie, Matt had a question in Q&A. Do you see
Oh,
that?
I don't see it, sorry.
Oh,
Sorry, Matt.
well, that's weird.
Yeah, that's weird. Yeah, I'm like, I thought I answered one and did that properly,
Yeah,
and
you did
now I
that
cannot
one.
see anything.
Mackenzie, can you look at the Q&A?
Oh
Do you
yeah,
get
I
it?
can see it. Yeah. Yeah, so right, is the change in the ratio of nitrogen 14 to nitrogen 15 as you go higher due to nitrogen 15 being selectively taken up, or is nitrogen 14 being chemically changed to nitrogen 15 inside organisms? Yeah, great question, Matt. Yeah, so that kind of goes into the whole chemistry, which can be really interesting with stable isotope analysis. So yeah, it's really that nitrogen 15 is kind of selectively held onto in the organism. So kind of the more chemical reactions that you have, the more likely it's kind of harder to break those heavier isotopes. So they end up staying more in the tissue, and then the lighter isotopes are excreted. So that's sort of why you end up seeing this increase in the ratio of heavy to light nitrogen kind of moving up. So yeah, so it's not chemically changed, it's just selectively taken up. Yeah.
So I did finally see the questions popping up. Sorry about that. So there's another question in the Q&A. So is dominance at a shared bait a good measure of abundance? Seems like it better reflects behavioral aggression than abundance, but perhaps a combination of the two, exploitative plus interference competition.
Mm-hmm. No, that's definitely, a good question. And I think, something that I talked with, we're kind of currently revising that paper right now. And that was kind of one of the very valid critiques that came up. And I think that, kind of shifting that more to numerical dominance. So yeah, using mainly the pitfall trap abundance data as a measure of ant abundance, just because that's kind of more passive sampling technique. And then the bait data, does kind of look at more behavioral aggression. And, I assigned that behavioral dominance score kind of based off of some, papers in the literature that use the, pitfall trap data and the bait data. But yeah, I agree. There's kind of a little more to that, that numerical dominance is kind of, I think, what's happening with the bait data. But yeah, I mean, good question.
So I am not seeing other questions, but are you?
No, not either.
Okay. Well, hopefully we've answered all the questions, and if we haven't, feel free to contact Mackenzie, and hopefully if you see her here and there on campus, please say hi. She's new; she definitely wants to meet more people in the department, and so I guess with that, thank you, Mackenzie. We definitely appreciated your seminar today, and yeah, I guess welcome to the department again. So, and thanks everyone for being here.